During aerobic respiration, carbon dioxide has liberated. During aerobic respiration, carbon dioxide has liberated. For performing the aerobic respiration, we needed germinating seeds. Actually, germinating seeds are not readily available. We have to do the germinating seeds. Okay. First of all, we have to take a handful of moong dal or a Bengal gram seeds and soak it in a whole night. The very next day morning, take out from the water and dries up, and just you tie in the cotton cloth and hang it in the window of in your classroom or maybe at home itself, whatever it is, the places are available, and we can make them the whole day. And the very next day, when you can open the pouch like a cotton cloth what you have used for tying the seeds, you can see that the seeds got germinated, right, and generally called as the sprouts, right. The germinating seeds, we need to take a handful of germinating seeds in a glass bottle or a plastic bottle. This is what a glass bottle we are taking, and inside the glass bottle we have taken the moong dal or maybe the Bengal gram seeds which are germinated, right. And take one glass tumbler and 3/4 of the glass tumbler we need to one third of the glass tumbler need to fill it with the lime water. 1/3 of the glass tumbler fill it with lime water. This is the lime water, right. Now we can close the glass bottle with a rubber stopper. Which can prevent the external or environmental air to enter the inside the bottle itself. So that tightly we can close the mouth of the glass bottle and leave it for 2 to 3 days. 2 to 3 days we have to leave this entire setup of experiment without disturbing, means undisturbed condition. We have to leave this entire setup in undisturbed condition. So when you leave that 2 to 3 days, after 3 days we can observe that the lime water inside the glass bottle what we have kept in the corner is turns into milky white. Whenever the lime water turns into milky white, nothing but the only carbon dioxide can convert the lime water into milky white, and there is no chance of carbon dioxide present in the atmosphere to enter the bottle and contact with the lime water, because the bottle has closed with a rubber stopper tightly so that air cannot enter the bottle. And how the carbon dioxide has produced inside the bottle, and we can see that the germinating seeds can performing the respiration and produce the carbon dioxide. This carbon dioxide has mixed with the lime water, and so that the lime water turns into milky white. This is the reason the carbon dioxide has liberated. We can easily confirm that during the aerobic respiration, carbon dioxide released. During aerobic respiration, carbon dioxide has released, and we can confirm that by doing this simple activity. Now we are discussing heat released during respiration. During respiration, carbon dioxide and heat liberated. And the previous activity we discussed that already the carbon dioxide has liberated, right? Now in this activity and how heat is released during the respiration we will discuss, right? For doing any activity we require some materials, nothing but apparatus needed. Here it is a thermos flask is needed, and a thermometer for knowing the temperature producing, and germinating seeds. Actually we need the same kind of setup, the one more setup also we needed, right? The same kind of apparatus we needed one more also. And here it is inside the thermos flask we have taken the germinating seeds. And germinating seeds already in the previous activity carbon dioxide released what we have prepared already, the soaking the seeds in the whole night, and the next day we need to tie in a cotton cloth and leave it for 1 day completely. After next day we can see that the germinating seeds. The germinating seeds we are taking in the bottle and observe the carbon dioxide has released. In this activity also with the same kind of seeds we are taking. Inside the thermos flask we have taken the germinating seeds. The same setup with the non-germinating seeds, right? And when you observe that a thermometer should be inserted through the splitted cork. This is the cork itself, and through the cork we can insert the thermometer, and the thermometer, the bulb of the thermometer should be in the middle of the germinating seeds. This is the one of the most important precaution. The bulb of the thermometer should be in the mid of the germinating seeds. Okay, and this kind of a setup, and after inserting the thermometer, just closely you can close the thermos flask mouth with a cork or a rubber stopper without entering any kind of atmospheric air or a carbon dioxide, any kind of things inside the bottle itself for the thermos flask. So that we have to tightly fix the cork after entering the thermometer itself. And this kind of a setup we need to be keep it aside for nearly 24 hours. Nearly 24 hours we need to keep it aside. The condition is for every 2 hours, we need to check the readings in the thermometer. We need to check the readings in the thermometer for every 2 hours. When we observe the readings for every 2 hours in the thermometer, we are observing that the temperature in the thermometer slowly raising. The thermometer readings are slowly raising, that we can observe, right? So whenever the temperature raising for every 2 hours in the thermometer, it's nothing but heat is releasing. The germinating seeds are performing the respiration. They are also be like a plant itself, and they perform the respiration. So whenever they are performing respiration, they are releasing the carbon dioxide as well as the heat also. So that heat we are measuring by the thermometer. So by this activity we can easily prove that heat released during respiration.